All right, let's unpack the article titled Prospective de Novo Drug Design with Deep Interactome Learning by Kenneth Atz et al., published in Nature Communications in 2024. One big challenge in drug discovery is inventing entirely new molecules, not just modifying existing ones. That's a massive task. A way AI can help is through something called chemical language models, or CLMs. These models learn to read molecules as strings and then build new ones by combining chemical pieces, kind of like writing sentences. But there's a catch. Many of these models struggle when it comes to context. They might know what a molecule looks like in isolation, but they're not great at incorporating biological context, like the shape of the protein binding pocket or how it behaves in a larger network of interactions. They also often require fine-tuning on task-specific data, which isn't always available, especially when you're working with a new protein target. That's where Dragonfly comes in, a deep learning framework that tries to solve these problems. It introduces an approach called interactome-based design. So, what does that mean? Think of the interactome as a giant network, a graph, that maps all known relationships between small molecules and the proteins they bind. Each node is either a small molecule or a protein, and edges represent bioactivity, for example, if a molecule inhibits a protein with high affinity. This graph is built from bioactivity data sets like CHEMBL, and here's the key insight. Instead of viewing molecules in isolation, Dragonfly somewhat learns from how compounds behave across this entire network. Now let's look at how it actually works, in simple terms. First, Dragonfly uses a graph transformer neural network, or GTNN, to encode the input. That input could be a 2D molecular graph of a known ligand, or a 3D structural graph of a protein's binding pocket. The GTNN analyzes the structure and transforms it into a latent vector, a kind of numerical fingerprint that summarizes the molecule or pocket. This vector doesn't exist in isolation. It's trained using bioactive ligand-target pairs from the interactome, so it captures not just structural features, but how those features relate to real-world binding behavior. Then, that latent vector is passed to a chemical language model built on an LSTM architecture. The LSTM decodes the fingerprint into a molecular string, generating a new molecule in the form of smiles, for instance. Importantly, this generation isn't random. It's conditioned on the structural context, the GTNN's encoding, which could come from a known ligand or a target binding site. The result is a molecule that should be chemically valid and structurally tuned to interact with the intended biological target. Dragonfly can also integrate drug-likeness, synthesizability, and even predicted binding affinity constraints. Dragonfly doesn't need fine-tuning. It's designed to operate in a zero-shot fashion, give it a new binding pocket or a known ligand, and it can generate viable new candidates immediately. That's a major leap forward, combining graph-based structural learning, language generation, and network-wide biological context into one unified model. That's it for today. Thanks for tuning in. Until next time, take care.